Temporal graphs can be used to model disease progression. Given such graph, representing a specific disease progression, the task is to identify genes that should be targeted by new treatments. Besides answering this question, we need to characterize underlying molecular mechanisms that contribute to disease progression. If we could identify subgraphs that interact closely at the early stage of the disease, and their interactions decrease as the disease progresses, then it may suggest that mechanisms needed for more normal biological functions are being disrupted by the advances of the disease. Conversely, if we could identify subgraphs that interact sparsely at the early stage of the disease, and their interactions increase as the disease progresses, then it may suggest that the disease is creating interaction between genes that are not present in normal conditions in order to involve processes needed by the disease. Potential novel treatments can then be developed to target these mechanisms. In our paper, we proposed a novel algorithm SDREGION, sparsifying or densifying region, that identifies subgraphs such that their density monotonically decreases or increases over time in a dynamic graph. We have shown that SDREGION is effective in real data and is scalable. We showed its effectiveness in the progression of lung cancer using 4 non-small cell lung cancer gene expression datasets. The effectiveness of SDREGION was shown through gene signature enrichment analysis and pathway enrichment analysis. From gene signature enrichment analysis, all regions were significantly enriched in biologically relevant gene signatures, indicating the effectiveness of SDREGION. Pathway enrichment analysis showed that SDREGION identified subgraphs that represent tumor progression mechanisms. Importantly, findings that were identified but were not highlighted in our observations may suggest potential novel mechanisms in tumor progression. While our validation shows great potential in biomedical field, SDREGION is generic and could be useful in other domains. Thank you for your interest. For more information, please refer to our paper.